my name is Angaroni Fabrizio from the University of Milano Bicocca, the Department of Computer Science. I'm going to present our previous work and our concept sheet proposal about the characterization of host virus molecular interaction via mutational signature analysis using ultra-deep sequencing data or raw NGS data. Okay, let's start. So in each patient, we can observe different swarms of different virus particle virions. Every virus particle has different genomes with different mutations. For example, in this case, we have three kinds of mutation, the green one, the blue one, and the orange one. And each set of virions with the same genome are called a quasi-species. A quasi-species is this mixture of quasi-species inside a human body with different genome can underlie the adaptive potential of the virus versus the immune system because it gives to viruses a lot of heterogeneity. Okay, how we can study these quasi-species inside the virus? The quasi-species inside the virus could be studied via RNA sequencing experiments. In this case, we have this population of virus, we sample it, and then we do a sequencing experiment. So we have our RNA reads. Then, aligning our reads with a reference genome, we can measure the number of mutations. For example, in this position, all the reads bears a C mutation. Instead, in this position, only two reads have a T mutation respect the reference. So for each mutation, we can define a variant allele frequency. So how much the variant frequency, the mutation is present in our sample. And we can grossly distinguish between two types of mutation. The clonal or fixed mutation, the mutation with like this one C, that have IVAF, for example, one, and then are present in all the virion of our sample, or the the mutation that has a low variant frequency, so are present only in few quasi-species of our sample. Obviously, if you study with consensus sequencing, by definition, you will lose all this in all of this information. This information is already employed in literature. If you there is a lot of pipeline that start from the SNV's profile of the sample, where we have the consensus variants, and here we have for each sample the intra-host or the minor variants, and we can test, for example, the drug resistance. For example, suppose that G is the mutation that bears the drug resistance with consensus sequencing, you will not find this mutation. Instead, with ultra-deep sequencing, you can look at this mutation and change, for example, the therapy. Or, for example, you can understand the resistance of an infection. In fact, during a chronic infection, there are a lot of mutations in your body. So you can observe a different heterogeneity of the quasi-species in our sample. Finally, other pipeline look at to investigate the transmission cluster and the contact between individuals. If the bottleneck size of transmission is very large, then you can simulate this process as a binomial process and try to understand what are the contacts between populations. However, we can say that most of minor variants arise inside a single host. So minor variants are the footprint of the inter-host mutational process, the process that currently working in a host. And to analyze this kind of footprint, but what are the mechanisms that are causing the observed variant? For example, here we have a G that have a transition to A. It is a single nuclear point mutation, single nucleotide mutation, and we know that could be represented as a substitution model, where we have the transition rate between the various nucleotides, and we can measure it empirically. And also in different organisms, for example, in human or other organisms, we know very well that the equilibrium of this reaction, for example, the transition T to C, is altered by some human process. For example, the Appleback enzyme that change the rate in the substitution model. How we can analyze mutational signature? Mutational signature mutation can be originate from exogenous factor or endogenous processes. All of these processes lead a characteristic substitution pattern, the mutational signature. So this I take for this slide from cancer work. For example, we do a bulk experiment, a sequencing of our cancer sample, and we obtain a mutational spectrum. You can see where we count the number of mutations that are C to T or T to G. And then we can decompose this process in the different sub-process that create this spectrum and the exposure, how much any process is active in that sample. Technically, this problem is a non-negative matrix factorization, where we start from the data D, where we have the number, the sample, and for each sample, we have the number of mutations that falls in a certain class. For example, here we have the C2A class and G2T class. And we have one row for each sample. Then with Monte Carlo approach, we can decompose this input data into different matrices. The first one is the signature matrix B. This matrix represents the composition of each signature. Here, you can see this, for example, here signature 1 has only substitution T to C. Instead, signatures 2 has different kind of substitution. And these are the footprint of the processes. And then we can also evaluate this low rank latent NMF matrix to understand how much a specific signature is present active in a sample. For example, for this sample, we have that signature 2 is more active than signature 1, for example. Okay. One important statistical thing about this kind of analysis is that you don't know in advance how many signatures are present in your data. So you have to do estimate the optimal number of signatures. This is made with different statistical tests. For example, here the best number of signatures is 10 because as the stable solution, and we have low reconstruction error for this case. Okay, so now let's see some result of this technique in SARS-CoV-2 and how we found some automated process in this virus. Okay, SARS-CoV-2 is a single strain RNA virus, and obviously it can mutate. It can mutate due the stochastic process of mutation because it's a virus, has a high replication rate and high error of rate, and can have both mutations that are single nucleotide variants or structural variants like index. This looking at the minor variants in particular are the natural background to study these molecular processes. So for SARS-CoV-2, we start with variant calling, we start with reference. We have our reads from the experiment. We do a 2C of the reads. For example, we take the reads away the reads with contaminants or primers, and then we align our reads to the reference genome. Then we do another quality control, secondary quality control on SMBs. For example, we take only the SMBs with high coverage. And then at this point, we have the SMBs profile of each sample containing both the clonal variant and both the intrahost variant for each sample. Then we start looking at briefly at informational landscape. First of all, we check that our SMBs are independent respect the number of SMBs is independent respect to coverage to be sure that we don't have coverage bias. So you can see a lot of mutation only because you have a lot of coverage. As you can see, there is no correlation, and we have sample with a lot of coverage with no mutation. Then we look at the distribution in our sample of the clonal variants, and we can see that minor variants are distributed as a power law distribution that is expected from the basic population genetics theory and indicate us the production of mutational processes that are happening inside the host. Finally, The last thing I want to point out that in this dataset of about 3,000 samples, most of the SNVs that we observe are intra-host. So with consensus sequences, you can keep only a small part of the SNV respect to the total. Okay. At this point, we apply our method for mutational signature analysis. So we start from the data. This is one sample, and this is the 6th category that we look, and we decompose it in 3 signature and the exposure matrix. The 3 signature in particular, we can relate them. The first one to ApoBec, that is well known in coronaviruses, that is working as cell mechanism defense, and the reactive oxygen species, ROS, we find, and the activity of other enzymes. Interesting, if we look at the activity of the sample, we see that, for example, the sample with high activity network does not have activity of signature 2, for example. And so in this using a clustering with k-means, we can find that there is 3 different clusters between patients. That indicates that the process of the patients are different. We repeat this analysis for other datasets, and these are the other 4 datasets, and the results are very similar, well, more or less the same. So we found all the 3 signatures and 3 clusters associated to signature. This cluster are the ApoBec cluster, another cluster, and a cluster, the second cluster, that has also ApoBec, also other, and also ROS activity. Okay. Then very important. Lastly, we want to quantify how this mutational process are selected. This mutational process gives an advantage to the virus. And so to do that, we try to understand the selection pattern in the various coding regions with the NDS analysis. However, there is a point. When you do this kind of analysis, or counting the non-synonymous substitution versus the synonymous substitution, you have to hypothesize that every nucleotide have the same substitution probabilities across the genome. But from the mutational analysis, we have an empirical proof that is not true, because patients belong to different clusters where the activity of the process is different. So there is a bias among nucleotides. For this reason, we developed a corrected NDS method to consider this the normalization due to these signature-related mutagenic processes. And then we evaluate the NDS. Interestingly, all the signature cluster have the NDS lower than 1, so there is evidence of purifying selection against signature-related mutagenic processes. And this can be, for example, ApoBec. So it could be explained because ApoBec is a cell mechanism defense in most in a lot of viruses. So it's possible that this is an example of how ApoBec purify certain virus. Okay. Summarizing the result on SARS-CoV-2, we look at an high resolution picture of SARS-CoV-2, and we found 3 different host-related processes. We also find that it's possible to clusterize and divide the patient, respect the activity of these processes, and the patient have an high heterogeneity. So there is different response in different patients. Finally, we also observe that most of these mutations are purified. So seems to be a negative selection, again, mutational process driven by signature. In addition, we develop also a pipeline for this analysis. This is a pipeline that you can find in Docker or Nextflow, where you can do variant calling with our pipeline, given the reference genome. And then you can also do a de novo discover of mutational signature in your dataset, or if you know or want to check if it is present a non-mutational signature in your data, you can do only the assignment of mutational signature. By the way, now I want only to say a brief something about our concept sheet proposal for HIV using the PANGEA dataset. First of all, we need to, in this moment, we are looking how to do intra-host variant calling for HIV because HIV is an organism with very, very high mutation rate and high probability of recombination. So in our test, we see that using a reference-based aligner with using the reference of HIV give us a lot of false positive. So we are currently testing to try to see the mutational landscape of intra-host variant HIV, no reference-based aligner like Shiva. Then we want to do a de novo discover in signature of the HIV. We know that ApoBec is already known in HIV, but we want to try understand the activity of ApoBec in different patients of HIV and try to find other causes of or other enzyme. I don't know. In particular, the attention will be to the drugs because we well know that in cancer, some drugs leave a footprint on our genomes. So maybe also studying the relation with metadata that contains drug treatment, we can try to understand if there is a fingerprint, a signature of drug in our data, and maybe try to estimate how each signature gives the probability to develop a no antiviral resistant mutation of HIV. Then, very important, we want to study with metadata and the cluster techniques. So we want to study if also HIV presents a heterogeneity of the host. So there we have different behavior of the molecular process of the host. It maybe could be they correlated to treatment, could be correlated to age, gender difference, or for example, different therapy, or the status of the patient, or the clinical stage of the disease. And we asked that maybe if that is true, signature could be used as prognostic test as we done in cancer. So I thank you for the attention. I want to thank the member of my lab, Alex, Marco, Chiara, Gianluca, Davide, Francesco, and Lucrezia, and our collaborator from Department of Medicine, Rocco and Daniele.